Good morning. Thank you for being here. Thank you, Loida. As Loida said, I'm going to give you a brief summary of the development of our research platform from our first cycle to where we are now. So as Loida was talking, in our first cycle, which ended in 2011, we had a very basic proteomics platform, which consisted in proteomic profiling with cell-detox MS, and either by fractionation in solution or in gel, we were characterizing proteomes, and we were working with subproteomes and full proteomes. By the end of the first cycle, we were doing protein identification, and we were starting to work with ingenuity pathway analysis for processing data. We're pretty successful the first cycle. We were able to do the mass spec technology transfer from University of Nebraska Medical Center. We acquired the LTQ, we acquired the DICE system, which are the most strongest platform from the core. And we offer workshops, seminars, and lectures to educate our scientific community, and we provided novel proteomics technology to 33 investigators. We were successful in fostering collaborations that resulted in grant awards, and 18 manuscripts were submitted for publication in our first cycle, of which 12 were published. We had some limitations in the terms of instruments. We had limitations with the maintenance and tech support for the LTQ MS. But our aim is focused on educating academic research. So we had a limitation that was very important for us, which was the different types of users we had. We had the new user to proteomics that is new to proteomics, expects training, has basically is totally new, has no idea or perspective on how the experiments last, how they're performed, how expensive proteomics experiments are. They're mostly overwhelmed. They were mostly overwhelmed by the large amount of data that we were generating for them. They require from us considerable assistance in working with this process data, which was something that we didn't expect at the beginning. We identified that validation from the users was a very limiting step for the publications, and that the publication process was very low. And this is important to us because NIH evaluates our success by the number of publications that are generated from the data that is generated from our lab. We also have a different user, which was an experienced user, which already knew about proteomics, already had done a basic characterization of proteome, and was interested in more advanced techniques as quantitative proteomics, post-translation, looking at post-translational modifications, read-up proteomics, and lipidomics. In order to target those two issues we had, or those two types of users, and make their research easier and our research easier, our part of the research easier, we enter a second cycle. We changed our name to the RCMI Translational Proteomics Center to represent more what we really do. And we had two aims. One, that was sort of targeting the more experienced user, where we wanted to expand methodologies by collaborating with international and national collaborators. And in particular, we wanted to include bioinformatic collaboration. And for the newer user, we wanted to increase our research productivity by providing a stronger training program. The first thing we did for the new cycle was move to this new facility here at the Comprehensive Cancer Center. We are grateful for the space that we have now, and when you see I hope that you all stay for the afternoon, the open house. You're gonna really like the lab. We're very happy. We have 1,200 square feet. We have an instrumentation room for the mass spec. We have a chemicals room, cell culture room, and we also have a cold room. So now we have space. We're happier, we can expand. So we've been expanding methodologies by acquiring new instrumentation. In the pamphlet that you received today, there is a copy of our guidelines, and on the end of the guidelines you have a picture and a description of all the instruments that we have in the lab that Yolanda Rodriguez put together very nicely. So I welcome you to look at that. But basically, what we did, we expanded our instrumentation for sample preparation for 1D and 2D electrophoresis. We got an off-gel fractionator for in-solution fractionator of samples. We have, and for preparation of samples for quantitation. We also I got a software for database search and to improve our analysis. And also we got instrumentation for validation. So we expanded our proteomics workflow, and now we have, regardless of the type of tissue, cell culture, body fluid, the samples are either fractionated or separated, either in gel, in solution, or even immunoprecipitated. We can do digestion in gel, in solution, and in beads. And we do a peptide extraction, cleanup, and then we do mass spec and validation. As looking at the areas of interest for the expert users that were requesting, we target for the whole cycle, we target the areas of quantitative proteomics, bioinformatics, post-translational modifications, and redox proteomics. For this first year, I am really pleased to say that we've been able to expand for quantitative proteomics. We've done experiments with iTrack, ICAT, and SILAC, and currently with O18 in Spain. And I'll explain a little bit further along. And we're starting collaborations. We're starting talking of collaborations with bioinformatics in mainland. The areas of research for us have always been focused on health disparity issues, which are as HIV and other infectious diseases, cancer, neurological and neurodegenerative diseases, and cardiovascular diseases. I'm just going to briefly talk about quantitative proteomics, which is what we've been expanding this year. So quantitative proteomics is for the discovery and target proteomic analysis to understand the whole system. There's relative quantitation methods, which are used to compare protein and peptide abundance between samples using isobaric tags. Absolute quantitation, we can use via SRM, using labeled synthetic peptides, and I'm not really knowledgeable about that method, but I learned recently that's something that's catching on in proteomics. And then label-free strategies are also available for relative and absolute quantitation. In our lab, we offer sample preparation and labeling methodology for SILAC, ICAT, iTrack, and O18. I just put a slide for you to look at. I can send you the presentation later on, but basically in all these methods you have 2 treatments or more than 2 treatments where you label the samples and you mix them together, and the samples are run on the mass spec at the same time. This way you remove a lot of factors that influence the intensity of your peaks, and you can do relative quantitation to one of your samples, which will be your control. So we are able right now to prepare the samples for these methods, and we outsource right now the mass spectrometry analysis and quantitation, because our instrument is not sensitive enough for this type of studies. In terms of the new user, this year we've spent a considerable amount on educating and training the scientific community. We have the RCMI website, which RCMI puts up for us and helps us keep it really nice. All the information about the guidelines, registration forms, contact information, and methods of operation are in that website. We have the core guidelines that you have it in your pamphlet, explains how we work, what we can do. We added an e-ticket system thanks to our CMI that allow us to organize and structure better our work and what we do for each user and how we record our work. We also included a methods timetable, and this has helped incredibly because the new user now can say, okay, I want to do a digest experiment, it's going to take me a month and a half. That's realistic. So when the user has realistic expectations, then we are able to meet those demands. When the user has very unrealistic expectations of they're going to give me a sample and next week they'll have results, then it creates trouble for me because it looks like I'm not meeting the expectation of the user. So the user is not pleased. So for us, it's very important that we discuss this with the user. We have also provided workshops, hands-on training, seminar series. We have a journal. We have a seminar series, and we also have a journal club that meets once a month for proteomics. We encourage everyone. We have most of our users are here. We encourage you to come over to our journal clubs because we discuss studies that have been published in proteomics, which give you the sense of what type of experiments you can do and what methodology is available there besides what we offer. And also hands-on training in the lab. For the first cycle, the one thing we really learned is that proteomics has a lot of challenges in the different steps that takes proteomics, from sample preparation, fractionation of the sample to make it easier or less complex, fingerprinting identification, and data analysis. In the first cycle, most of our effort was here, learning the techniques, working with the experiments, optimizing that area, but we realized that we had to spend a lot of time with the user in these areas, the sample preparation area and the data analysis area. So we laid out, for the second cycle, we laid out a very structured method of operation, okay, in which we do a first consultation where we do the experimental design. We do the second step with sample preparation and quality assessment. The third and biggest chunk of the method is the experiments. Then we have data analysis and validation and publication. So we spend right now, this second cycle, we put a lot of effort in consultation, sample preparation, and data analysis with the user. So I'm going to give you an example of how a regular experiment would run. So for the initial consultation visit, we discuss the hypothesis, the research questions, the objectives of the researcher. We explore options in proteomic analysis, the services that we provide, and if you can really do what you want to do with the services that we can provide. We have to look, I have to do a lead review and design a study depending on your system. If you use bacteria or tumor cells, or if you're looking at CSF or blood or urine, or if you want to look at the whole proteome, or you want to do target PTMs analysis, I have to, for sample preparation, I have to study to be able to give the best advice. We discuss collaborations and fees. We discuss the user's guidelines, the registration form, the timetable, and the kind of experiments. I spent at least 2 meetings in this section. I also talk a lot about sample preparation, and we make sure that the person is lysing the cells right, extracting the protein right, doing the quantitation right. Sample preparation is the greatest challenge in protein discovery and analysis. It's absolutely essential that you determine what's the optimum way to prepare your sample empirically. The objective, depending on what you want to do, if you want to do the complete proteome, a subproteome, or if you're purifying a recombinant protein, this is all things that, depending on the answer, the methods change. Or sorry, the preparation of the sample change. We also, if possible, if the person has enough sample, enough quantity, we try to do 1D gel for quality assessment of the protein before we even start the experiments, because these experiments are pretty expensive. So, ¿ya tengo cuánto tiempo? ¿Me pasé del tiempo? The strongest platform, so for experiments, I'll give you an example of the strongest platform we have, which is the DICE platform, which gives you differential expression of the proteome, and the MS gives you the identification of the proteome. So expression, identification, plus analysis that puts it all together is what gives us the understanding of the protein interactions going on. So I'm just gonna move through these quickly, but basically the DICE basics is a 2-die or 3-die, allows you to also use 1 or 2 samples with different proteins that are separated on the same gel. Since the samples are pre-labeled and mixed, you can use a internal standard to normalize throughout all your gels. This is a basic workflow. This takes about 2 weeks for the preparative gels, and then 2 weeks for the, 2 weeks for the analytical gels, and 2 weeks for the preparative gels. I'm not gonna go through that. Okay, so once we run the gels and we collect the bands, either from 1D gels, 2D gels, or even in solution, we take the protein, we digest them, they go to the mass spec, they are identified by database searching, and that's what gives us identification. So for this cycle, we've expanded our capabilities for database search by buying the Proteome Discoverer and the Mascot license. So now we have Sequest and Mascot, which are major search engines for database search that can be run from the same interface, which is Proteome Discoverer. It's a software that it's provided by Thermo, or it's sold by Thermo, to be able to process our raw files that come from the mass spec, which is also from Thermo. So once we do the analysis, that's where our analysis ends. The report, we report the results to our investigators, either in a report or a summary of generic methods section, so that it facilitates the report manuscript preparation. For 2D DICE, we export tables from the software that gives you the spot numbers, p-values, expression, fold changes, and the protein ID tables with all the protein identified that pass the exclusion criteria, proteins that have more than 2 peptides, and we have a scaffold or a Proteome Discovery viewer that we can send our users, and they can see the files themselves of the data, which has proven very useful for us, especially with Scaffold, since Scaffold is a very visual program with different colors for upregulation and downregulation, and we've noticed that for users it's really advantageous to use that software to understand their data. However, we're now our next obstacle that we've been trying to work with is what we do with all this data. This is our problem now. Once we're done with all the experiments, the investigator has a really big issue and a really big set of data on their back. The data that's generated from these studies is really overwhelming, and nobody at this point is expert in all the proteins that are in a proteome and how they interact. So it's really hard to put all that together and understand it. So in the way that we've been working is that the investigator is responsible for doing the lead review. Once they get the data, they have to sort through the proteins. So that was really overwhelming for the user. They had to organize and integrate the data relative to the system of interest. They had to do further bioinformatic analysis, validation, and preparing data for reports and manuscripts. And we've realized that some of our users are not ready to do this by themselves. Okay. So we give them resources. There's free resources on the web, and we also work a lot with Ingenuity pathway analysis. So basically, I've been including the Ingenuity pathway analysis in the reports, which in the first cycle was not part of the report. So doing this, what I do in Ingenuity pathway analysis is a basic a core analysis of the data set, and then depending on, I call the user sits with me, looks through the data, and if they're interested in further looking at specific proteins, then I give them the access to the Ingenuity pathway analysis software, and I sort of lead them, but they can work on the software by themselves. So Ingenuity pathway analysis, for those of you who don't know, you can input expression, protein identification and expression, and looking at the knowledge base, which include NCBI, clinical trials, all sorts of data sets available on the web. It gives you an idea, an idealistic pathway of how the proteins that you found upregulated and downregulated may interact with other proteins in known pathways. So it's proven very useful for our users to be able to at least cluster proteins in certain function pathways and understand better their data. One of the other problems we face is validation. Once the user receives all this data and do all this analysis, they have to validate those changes with direct monoclonal antibodies, ideally in a large number of samples and in different samples that were used for the experiment. This also has proven to be a big task and something that we've been trying to help users for this cycle. So our new approach for this year is to assist greatly in manuscript preparation to the users. We are going to require users to provide proof that they have the funds to do the validation before they start the experiment, because once we start the experiment, we run the experiment, we do the analysis, and we turn the data to the user, the validation is critical for publication. And if that's not done, then the publication doesn't go out, and then that's the way we get evaluated. So we're trying to make sure the user knows that they have to validate, has the money to validate, give them, we make sure that they know a timetable, have a realistic idea of the timetable. We're trying to outsource bioinformatics expertise and putting those bioinformatics in communication with the user. That has also helped a lot. We're trying to, also in this new cycle, trying to access a new mass spec instrument, that's the Biomolecular Center. And sorry, these are switch. And we're also trying to include support for validation techniques. So even if the user maybe does not have all the funds to do the validation or doesn't have the proof to do the validation, we are willing to work a collaboration or some sort of assistance to try to get these studies validated and published. So that's our aim, our real aim for this second cycle, is to really support the user into publication. So with that, that's the end of my talk. If you guys have any questions or would like to say anything. Okay.